So now we get to the tough one. Is that I'm not absolutely sure I'm going to be able to see what I need to see on this, because some of these have not been stained quite as darkly as they could have been, and sometimes it's just really tricky to get it to work. Even in lab, I tell people, sometimes it takes a couple of tries.
You need to tell them what you're doing, though.
I am, well, I wanted to make sure I could find something first. I'm looking at Clostridium tetani.
Yeah, but what is it?
Oh, it's bacteria, sorry. Everyone should thank the director for keeping me on track here. Now, right now, I'm off on this, I'm looking at the label. That's not going to really help me. Okay, there's the edge of the cover slip. And there's stain. So this is telling me that I'm looking at, now here's the funny part. This is probably 300,000 or 400,000 bacteria. Because at low power, all I'm doing is looking at little clumps of them that have been stained. But at least I found them. So I'm going up to middle power. I'm getting them in focus. I'm going up to high power and getting them in focus. Now here's what I always tell people. You look at a smear of these guys and figure out what is the smallest repeating shape you keep seeing. And unfortunately, there's two shapes here, but only one of them is the bacteria. The dark dots are little granules of stain, unfortunately. But see these little streaks here? Streak, streak. Let me play with the focus a little bit. Streak there, little streak there, tiny little streak there. Smallest repeating shape, streak, streak, streak. These are bacilli, which is a very particular shape of bacterium. Sometimes they string together, but this shape is reliable for this type of bacterium. And that's, as I said, if you're drawing bacteria, all I want you to do is give me a shape. Every once in a while, if the stain picks up, you'll see a dot. They don't have nuclei, but they do have some structures inside that will show up on the stain. But most of the time, it just gives you a shape. And for Clostridium tetani, that's perfectly okay. Okay, so I'm going on to the next one. I'm gonna take my slide and make sure that it's nice and clean. I'm using a piece of paper towel to get the dust off of it. Normally I would use my shirt tail, but I just want to be pull my shirt up to do this. Back down to low power. Get the edge of the cover slip in focus. Got to take my glasses off. There's the edge of the cover slip. Let's see if I can find something stained. There's a little dot of red. That's weird. I am not terribly confident about this. There's a decent amount of stain, but the way that the stain looks, it looks like that's all I'm really looking at is stain. Well, that dot gives me something to zero in on. That dot is definitely not bacteria, it's too big, but it may have bacteria around it. So I'm gonna go to middle power. Get that dot back in focus. Oh, I'm starting to think there are bacteria around it. Get the dot back in focus. Now we wind up with running into problems I have because I'm an old person. And my eyeballs are full of crap. And it lights up looking at a background like this. And it's very difficult to focus on little things when I've got all these strings of tissue in my field of view.
Yeah, you had it in better focus before.
Okay, I'm messing with it. Okay. This is where it starts to get tricky. Imagine that this is a cloud of smoke. And someone said, well, you know, these are all particles, and the particles are all the same shape and size. Pick one out and draw it. So the trick here is to look into the spaces for shapes that repeat, because here they're all piled on top of one another and you can't really see them. And this is very tricky to get to work on this screen. So do your best, try to pull some of these things out of the spaces and draw me a shape. But that's about the best you're going to be able to do with this. This is why I only have you do one bacterium. Bacteria are hard. Okay, so that was Klebsiella pneumoniae. The next one is Neisseria gonorrhea. Some of you already know what disease that causes. And let me see what we got. That is a crack. I don't know, there's the edge of the cover slip. Well, we got some nice stain. This is very similar to the first one. The trick here, again, look for the smallest repeating shape. And I'll play with it so to help you here. If you look in this one isn't stained that dark, so you can actually see the bacteria. There's probably 12 or 15 bacteria in this clump. And my question to you is, so what shape are they? One of the problems you get is the bacteria kind of settles around them, so they're kind of sitting in a red puddle. But they definitely are of repeating size and shape. You can sort of see them in the big clump, but it's harder. The more stain there is, the harder it is to make the bacteria out. But on the edges, you can see them. That's why I like the lighter ones where you can see. The really light pink is settled stain. The dark things are the bacteria. So there's definitely repeating shapes there. Weirdly enough, if you're looking at stuff through the microscope, this thing that I'm doing where I'm twiddling the focus back and forth actually helps you to see things through the microscope as well, because of the way your brain processes movement. It'll pick stuff up when it thinks it's moving, because you change the focus. Okay, on to the next thing. The next thing is Streptococcus pyogenes. The strep part you probably are familiar with. Down to low power. There's the edge of the cover slip. Let me get that in focus. Get out into the, oops. Make sure I get this clean. I don't want to be looking at dust. Out into the middle here. See how much difference that made? Now here we got a nice big blob that's been very well stained. And again, we're probably looking at least 500,000 bacteria in this blob. They are very tiny. Now we're up to high power. No, we're not. Nope, we're at middle power still. I was gonna say, bacteria are too small. Here we are at high power. These are pretty easy to make out. Well, they are through the microscope. They're not showing up on the screen as well as I would have liked. They're strings of, every one of those strings is multiple bacteria. Oh yeah, you can see them. Like, look at this string here. That string, pretty obvious what the members of the string are. Very commonly, bacteria will reproduce and not completely let go of one another when they make copies. This is about the best prep I've seen on any of these slides. It's very, very easy to make out the bacteria and to make out what shape they are. So that worked out fairly well. I think we can go on from Streptococcus to the next one. The next one is Treponema pallidum, which causes syphilis. And this is a little unusual in that, back down to low power, is that what we have on the slide is a section of tissue. And what's in the tissue, once I get it in focus, the tissue's been stained this kind of ugly yellow, and the bacteria have been stained with a stain that's kind of grayish. It actually has silver in it. So at this power, middle power, you can sort of see the grayish, but you can't really see what it's doing. And at high power, okay, there's a little bit too many bacteria right where I'm looking. I want to get someplace where they're more spread out. Okay, so, see this dark streak? There's a dark streak there, dark streak, dark streak, dark streak, all over the place. You see these kind of S streaks are slightly curved but double curved. That is typical of what's called the spirillum bacterium. And the tissue is just full of them here. And they've picked up this dark grayish stain against the kind of yellowish reddish background. So again, draw me the shape, get a shape down for one bacterium. And this is Treponema pallidum. And those are the bacteria, at least those are the ones I'm going to show you. We had more of them in the box, but I tried to pick ones I knew I could make show up on the screen. So that's it for the bacterial part.